This video is the first video on linkage. So linkage is looking at how inheritance patterns are different when we have two genes that are close to each other on the same chromosome. So first we're going to revisit Mendel's dihybrid cross. Mendel did this cross with two traits, and he came up with this principle, the principle of independent assortment, that essentially says that genes are independent from each other, that they assort independently from each other during the formation of gametes. So here is one of the crosses he did. You have a individual who's heterozygous for two genes, that is a dihybrid in the F1 generation. And we have discussed in detail how you can get four different types of gametes from this particular dihybrid. And so those four are all equally likely because big R could either be with big Y or big R could be with little y, and those two are equally likely. Now, the reason why independent assortment is observed is usually because those two genes that we are looking at are on different chromosomes. And so chromosomes are assorting independently during meiosis and during the formation of gametes. So what's shown here is the independent assortment of chromosomes, which have these two genes, the A and the B genes, on them. So here we have the formation of sister chromatids. During S phase, you form sister chromatids. And as you enter into meiosis 1, you would line up the chromosomes into tetrads, and the homologous chromosomes line up with each other. You can see here that this individual is a dihybrid. So one of those homologous chromosomes has big A, the other has little a. Same with B. One has big B, the other has little b. Now, when those chromosomes line up together to form tetrads, it's really random whether big A is on the same side as big B, as is shown here, or whether big A is on the same side as little b, and these two get segregated together. Whichever way they segregate, you would pull those chromosomes apart, and at the end of meiosis 1, you would end up with either the two dominants together in one cell and the two recessives together in the other cell, or else one dominant and one recessive together. Incidentally, in this particular example, we're simplifying our analysis by ignoring the presence of crossing over. During meiosis 2, the sisters would pull apart, and at the end of meiosis 2, you would end up with gametes that had the two dominants, the two recessives, or one dominant and one recessive. And all of these would be equally likely. Now, when we look at a situation where two genes are together on the same chromosome, things are going to be different because you don't get them assorting independently. So I'd like to draw out what we would expect to see if the A and the B genes are together on the same chromosome. And again, we're going to assume that there's no crossing over to simplify things. So in this particular case, we're going to look at a situation where we have a dihybrid. So this would be a big A, big B, little b, just as we've been discussing. And in this case, the two dominant alleles are together on one homologous chromosome, and the two recessive alleles are together on the other homologous chromosome. After we undergo S phase, you would have duplication of each of those chromosomes, and the two big A's would be together on sister chromatids, and the two big B's would also be together there. Same with the recessives. If you separate these two homologous chromosomes during meiosis 1, so this is a tetrad and you would pull them apart, one of them goes to one cell, the other goes to the other cell, you would end up with two cells, and they look like this. The dominant alleles were segregated into one of the cells, and all of the recessive alleles were segregated into the other cell. If you then go through meiosis 2, You would separate those sisters from each other, and you would end up with a situation where you have only the two dominant alleles together in those first two gametes, and only the recessive alleles together in the second two gametes. And so this is a situation where you have two genes that are close together so that they don't get separated by crossing over, and you only make two of the gamete possibilities. You don't end up making these two other combinations that we have previously expected. So I'm now going to introduce some new terminology that helps us follow which genes are together on a chromosome. So we are going to use parental type gametes and recombinant type gametes. These terms tell us about which A and B alleles are together on the same chromosome. So here you have this cross. It's a cross between parents that are dominant for two genes and one that is recessive for two genes. And if you think about a situation where these two genes are together on a chromosome, you'll realize in this case that big A is together with big B and little a is together with little b on a chromosome. When you get fertilization, you form this dihybrid individual, but this dihybrid is organized in a particular way so that there are two chromosomes. One of them has big A and big B, the other has little a and little b. When this individual forms gametes, we're then going to call the types of gametes that had the same allele combinations back in the parent generation, we're going to call those parental type gametes. So we have big A together with big B is one parental type, and here it is again, and little a together with little b is another parental type. Any allele combination that is different from what we saw in that previous generation, we will call a recombinant gamete. And those types would be big A with little b and little a with big B. Now, it's really important to realize that it's not always the two dominants that are together and the two recessives that are together. So here we have a situation. This is going back to the cross we talked about earlier. And in this particular case, we had a parent that had one dominant allele and one recessive allele. And this was a true breeding individual. So they make a gamete with big R and little y. And the other parent is little r with big Y. And when you make a dihybrid in this cross, the recombinant and the parental gametes are different. So here, the parental type gametes are the ones that came from each of the parents. Big R with little y and little r with big Y would be the two parental type gametes, and the recombinant type gametes would be the big R and big Y and little r with little y. R stands for recombinant, P stands for parental. Now, it can get pretty complicated keeping track of which are parental types and recombinant types. And so we use a particular type of notation, which we call linkage notation, to indicate which genes are together on the same chromosome. So here is a situation where you have two genes on the same chromosome, the A and the B genes, and these two chromosomes are homologous to each other. This individual is a dihybrid, heterozygous for two genes. And I just want to point out that there are two ways that this dihybrid individual could have their alleles arranged on these chromosomes. So either the two dominant alleles could be together on one chromosome and the two recessive alleles on the other chromosome. This would be called coupling or cis configuration. Or we could have a situation where the one dominant allele is together with the other recessive allele and recessive with dominant. This would be repulsion or the trans configuration. Most of the time in class, I will write out the arrangement of the chromosomes just as is shown here. One thing to be careful about, sometimes I see students putting the two alleles of one gene, big A and little a, together on one chromosome. That would never happen. You always have the two A alleles on separate homologous chromosomes. A further way to write these two chromosomes, the arrangement of these chromosomes, is actually just to put one line between the two, which indicates that the big A is together with little a with big B, or else even just put a slash. So here we're organizing the two alleles that are together on one chromosome together before the slash, the other two after the slash. Okay, so now we're going to revisit this cross that we've looked at before, and I'm going to ask you guys to just pause the video for a second and try to rewrite this parental generation going through to the F1 generation in linkage notation. All right, so this is what your linkage notation should have looked like. This true breeding individual who is dominant for both genes, we're now going to rewrite it with big A, big B slash big A, big B. The recessive, little b slash little b, and each of these gives the gametes that are shown. When those two gametes come together in fertilization, we form the F1 generation, and these arrangements are going to stay in place. So the big A will stay together on a chromosome with big B, and little a will stay together on a chromosome with little b. Next, we'll move on to looking at the gametes, the progeny from the F1 generation. So normally we would have a dihybrid individual. We can form 4 types of gametes, and the two parentals are on the left, the two recombinant types are on the right. When the A and the B gene are very, very close to each other on a chromosome so that there's no crossing over between them, it turns out that you only get the parental type gametes. You would never get recombinant gametes. Now, occasionally, recombinant gametes are observed, and that is if the genes are a little bit further apart from each other. They can be separated from each other via crossing over. So if you had a situation like this, where the two dominant alleles were together on a chromosome, the two recessive alleles were together on the other homologous chromosome, the only way that you can ever separate the big A from the big B is via crossing over. So these would be the parental type gametes here, and the only way you can get recombinant type gametes is if you get a crossover between the A and the B genes. That's shown here with these two non-sister chromatids, and that in fact can produce a new allele combination, which would be a recombinant type gamete, big A with little b or little a with big B. Now, when we investigate linkage, we are going to use this type of cross pretty regularly, where we produce a dihybrid individual and then look at the parental and the recombinant type gametes that result from that individual. Now, of course, you can't observe phenotypes in gametes themselves, and so we need to actually make a cross in this F1 generation in order to observe what we're getting from this individual. What we will always do for this type of experiment is we will cross the F1 individual to a tester. So a test cross, if you recall, is a cross to a homozygous recessive individual, and that individual will always produce only one kind of gamete, little a, little b. And so we would get these parental type gametes, recombinant type gametes, and this terminology can also be extended then to the F2 progeny of this cross with the tester. Each of these parental types are going to get that little a, little b from the tester, And this allows us to observe exactly what type of gamete came from this dihybrid individual. So the parental type progeny would be listed here. These are listed in linkage notation, and the recombinant type progeny are listed here. So to summarize this part, we're going to be looking at these different cases. We've looked in the past at unlinked genes, and unlinked genes, we would expect all four types of gametes to be equally likely. And therefore, if we would do a test cross, we would find that each of those four possible F2 progeny would be present in 1/4 each. If you had completely linked genes, you would not get any recombinant progeny, and so all of the F2 progeny would be split between these two types of parental F2 progeny. Next time, we'll look in more detail about situations where we have some linkage between two genes, but they're not completely linked. They're not so close that you never observed crossing over. So this brings us to our actual definition of linkage. The definition of linkage is not what you might expect, that two genes are together on one chromosome. We're going to define linkage based on a functional test, and this is a cross between a dihybrid and a tester. So for unlinked genes, we expect these A and the B genes to assort independently of each other, and you would get 1/4 of each of these types of progeny. Another way to describe this is that you would get 50% parental progeny in the F2 generation and 50% recombinant progeny. Our definition of linkage is that linked genes produce less than 50% recombinants. So here we have the A and the B genes are together on a chromosome. Here's a dihybrid individual in the F1 generation crossed to a tester. And here we have parental type progeny. For these ones, we know that the alleles that are together on a chromosome are more likely to occur than the recombinant type alleles. So we have big A and big B tend to be inherited together. Little a and little b from the F1 here tend to be inherited together. And we'll get more than 50% of the progeny being parental type and less than 50% of the progeny being recombinant type. So to summarize, when we have genes that are linked, that means they are fairly close on the same chromosome. The frequency of recombinant progeny is less than 50% when we cross a dihybrid to a tester. The way that you can get some recombinants is by a crossing over event between those two genes that are being investigated. So crossing over is the way that we can produce recombinants when we have linked genes. When we look at unlinked genes, we expect all four of these F2 progeny types to be equally likely, and the frequency of recombinants should be 50%, since there are two of those four possibilities that are recombinant. The way this usually happens is because these two genes are on different chromosomes, and recombinants are then produced by independent assortment of chromosomes. There is another way that you can get recombinants produced with unlinked genes, and that is a case in which the two genes that are being investigated are on the same chromosome but are very far apart. When you have this kind of situation where two genes are so far apart, the chance of getting crossing over or even multiple crossing over events between those two becomes more and more likely. And at some point, big A is no more likely to be inherited with big B than it is to be with the allele that is found on the other homologous chromosome. So therefore, You can actually get recombinants in unlinked genes. This would give you 50% recombinants, also by crossing over. So just to summarize this, if you have genes that are linked, they must be on the same chromosome. However, genes on the same chromosome, not all of them are always linked to each other. Even though they are physically on that same piece of DNA, nevertheless, when they get far enough apart, those alleles are not preferentially inherited with each other, and at that point, they would be considered unlinked.